Greetings to you all in the name of God. Today, in this video lecture, I will be discussing you about the cardiac markers. This topic is included in MBBS biochemistry syllabus of MBBS first year students. So let me go to the board so that I can handle the topic nicely. So here, as the board is quite visible to all of you, that the topic I am going to discuss today is the cardiac markers. Now, what are cardiac markers actually? The cardiac markers are the substances which are released in the blood after the heart attack. After myocardial infarction or a heart attack, these substances are released into the blood. So, if a person is having chest pain and he rushes to the doctor, and by the time he reaches to the doctor, his chest pain is over. So, how the doctor will detect that what had happened to him? In order to check whether the chest pain was due to a minor or a major heart attack, the doctor has to go for the blood test, and these are the markers which are found in the blood, so that the doctor is able to know that the person suffered from a heart attack because of the chest pain. So they are the substances which are released into the blood after the myocardial infarction or heart attack. So it is a diagrammatic representation of what I am explaining. The cardiac markers are released into the blood after heart attack by the damage of the heart muscles. So these are the blood. Now, cardiac markers are substances. When I'm saying substances, so they can be proteins, they can be enzymes, or they can be hormones. They can be enzymes, proteins, or hormones released by the ruptured muscles of the heart, and then they are released into the blood vessels, and they are detected in the blood serum. So that's why we call these cardiac markers as the serum enzymes. Now, they are the important cardiac markers. That is cardiac troponin, which is a protein, not a hormone or not a enzyme. Creatinine kinase CK2MB, SGOT, and AST. I will be discussing you about the full forms of all these enzymes and proteins when I will and when I will individually discuss these concepts. Then another cardiac marker is myoglobin. Lactate dehydrogenase is it's an enzyme. Now they are the common cardiac markers which are being utilized today for detecting myocardial infarction. The most commonly used cardiac marker today is the cardiac troponin. But along with these cardiac markers, there are some new cardiac markers as well, and these are also detected sometimes if the doctor is not able to detect the regular cardiac markers in a rhythmic way, then these markers are also used by certain doctors. That is ischemia, modified albumin, glycogen, pyrophosphorylase, pregnancy associated plasma protein, brain neuroretic peptide, and myeloperoxidase. They are again used as a new cardiac markers. So I am not going to discuss these because these are less used, but mainly these cardiac markers are used to detect the heart attack. So I will be discussing each one of them one by one. So in coming along with the cardiac troponins, it's not a hormone, it's not a protein, but it's not a enzyme, but it's a protein which is found inside the blood in high concentration after the heart attack. So it is mostly used nowadays. This protein is mostly used nowadays as a cardiac markers. Now it is a non-enzyme protein, which is not an enzyme, required release from the cardiac muscle after the myocardial infarction. Now this troponin exists in two forms. Structurally, there are differences in the polypeptide chain, and that is troponin I and troponin T. I am not entering into the details of its structure because we are not concerned with that. These two cardiac markers are used, troponin T and troponin I. Now, after the heart attack, the concentration of troponin T, it increases after 24 to 48 hours and returns to the normal level after 3 to 4 days. And likewise, the troponin I, it is also there. The concentration of troponin I is maximum after 72 to 100 hours, and then it returns to the normal level by 7 to 12 hours. So this is the case with the troponin uh, cardiac marker. Now another cardiac marker, which is an enzyme, it is creatinine kinase. In short, it is written as CKMB2. It's an isoenzyme, and the isoenzymic form, that is MB2, is used usually as a cardiac marker. Now this is the main function of this enzyme, which is being catalyzed in the body. It converts creatinine to creatinine phosphate and vice versa. with the use of this enzyme, with the use of ATP, which is converted into ADP. That means the phosphate is taken from here. So this enzyme is regularly converting the creatinine into creatinine phosphate. Now, this enzyme is the first enzyme to be found in the serum after myocardial infarction, and it is also the first enzyme that returns to its normal level after the heart attack. So, this is the first enzyme which is detected in the blood serum, and it is the first enzyme which is returned to its normal level after the cardiac attack. Now, there are three isomeric forms found by the two polypeptide chains, that is M and B. In the structure of this creatinine kinase, there are two protein chains, protein chain M and protein chain B. The protein chain M is visually found in the muscles, while protein chain B is found in the brain. So, there are three isomeric forms which are represented as CK1, CK2 and CK3. The CK1 consists of both the chains of consisting of brain BB, that is found in the brain, and the CK2, which is the important cardiac markers, it is available in the heart. Now CK3 is consisting of both the polypeptide chain, that is M, and it is found in skeletal muscles and also in the heart muscles. But we make use of the CK2 as a cardiac marker because it is exclusively present in the heart. Now, the activity of creatinine kinase is mainly found in skeletal muscles, brain and heart, and it is not found in liver and RBC. So, if we want to detect anything wrong in skeletal muscles, brain or heart, especially heart, we go for the creatinine kinase as the cardiac markers. Now, another concept is the concentration of CK1 increases in newborn babies, that is neonates, when they suffer from certain brain damage or head injury or weight loss, then the concentration of CK1 increases. Now, concentration of CK2, this is the second isomeric form, it increases in the blood in a normal human being after the heart attack, that is myocardial infarction, and all kinds of dystrophies and myotrophies if the person is suffering from. In a general rule, we notice that the activity of the creatinine kinase is inversely proportional to the thyroid activity. Now, if we quick have a look on the concentration increase and fall to the normal level, that is CK2MB increases in the blood after 10 to 12 hours, and it returns to the normal level after 2 to 3 hours. Now, the third enzyme which is used as a cardiac markers is AST, is also called as SGOT. Now, this enzyme serum AST, that is aspartate transaminase, and it is also called as SGOT, serum glutamate oxaloacetate transaminase. These are two same names. Enzyme commission number is 2611, and this is the function that this enzyme perform. It convert in our body, in our cells, this enzyme converts aspartate, and it combines aspartate with alpha glutarate and convert it into oxaloacetate and glutamate. That's why the name is given serum glutamate oxaloacetate transaminase. So it transfers the amino group. Now this enzyme AST or SGOT works along with a coenzyme pyridoxal phosphatase. Usually it is there. So this is the mode of functioning, and this is the function of this enzyme which is performed in our body. But this enzyme, when released in high concentration after the heart attack, is used as a cardiac marker. This enzyme is found in the cytosol and mitochondria, that is cytoplasm of the heart muscles, and normal range is 8 to 20 unit per liter. The concentration of this enzyme increases after myocardial infarction, muscular dystrophy, and etc. So that's it. Heart attack is the main source from where this enzyme is released in high concentration. That's why it is detected. Its concentration increases when I say upper mark. These two arrows indicates increases. So concentration of this enzyme, that is AST, increases after 6 to 12 hours and reach to the peak in our body, in our blood, by 24 to 48 hours, and then finally it returns to the normal level up to 4 to 6 days, or 4 to 6 days. Now this is all about serum AST, that is aspartate transaminase. When we speak about the LDH, it is another cardiac marker, and the full form is lactate dehydrogenase. Now this lactate dehydrogenase its concentration increases in 12 to 18 hours after the heart attack, and its peak reach in the blood serum after 48 to 72 hours, and within 6 to 10 days it returns to the normal level within the body or within the serum of the blood. Now these are the different enzymes, and when we say in a flipper pattern, it means this enzyme LDH is also an isoenzyme exists in two or three different forms. LDH1 is one of the form and LDH2 is another form. But in acute myocardial infarction, that is severe heart attack, the concentration of LDH1 is more than LDH2. Usually LDH2 is more. This pattern of occurrence of LDH1 more than the LDH2 in the blood serum is called as flipper pattern. So these are the different enzymes. I'm going to discuss once more. Uh, when we say cardiac marker, the cardiac markers are the substances which are released in the blood after the heart attack. Important cardiac markers are cardiac troponins, creatinine kinase, AST, SGOT, and myoglobin and LDH. Myoglobin is also there. Myoglobin is the protein which is present in the I didn't discuss about. Myoglobin is a protein which is present in the body. It stores oxygen, and it is also used as a cardiac marker. It is also released after the cardiac attack. There are certain new markers also. So cardiac troponins I have discussed, creatinine kinase I have discussed, then serum AST I have discussed, LDH I have discussed. Myoglobin I have not written, but I have discussed it. Now this is a chart which will be helpful in clearly visualizing the things. In the left side I have written the names of the enzymes, that is cardiac markers, and then abnormal activity is detected within this time, and maximum rise in the blood serum it is found in this row, and the time returns to the normal in this row. If we want to deflect this chart in the form of a graph, then it is here. This graph clearly shows that number of days or hours are increasing below and then activity is increasing up. So these are the different cardiac markers which are shown with the help of different graphs. So hope this video is helpful to all of you. Thank you very much.